I wanted to point out first that we have looked at basically the neuronal part of the brain so far at this course, and we wanted to also incorporate a bit of glial studies and neuron-glial interactions in this course, because it's becoming an emerging new topic in the field of neuroscience. And this is where my research group has been very active recently. So I myself started already looking at glia in 1990s with in vitro studies, but then we came back to this topic some 8 or 10 years ago. And I will now first introduce you to glial and especially astrocyte biology and neuron-glia vascular interactions, like from the biological perspective. And then I will move on to modeling astrocytes and neuron-astrocyte interactions in synaptic plasticity. So what are glial cells? Glial cells are non-neuronal cells in the central and peripheral nervous system that contribute to various functions in the developing and adult brain. And what is important to notice is that as the nervous system through evolution, when it developed from this kind of diffuse system to more like centralized form, like we have now in rodents and mammals and humans, so then the neurons were joined by the appearance of the supportive cells, so to say, this neuroglia. And also, it is important to note that the number of glial cells and their complexity especially increases with the overall complexity of the nervous system. And although we know quite little, at least in my mind, about the glial contributions to brain functions, they were discovered already very early in 1856 by a German pathologist Rudolf Virchow. So it is important to know that there are different types of glial cells in the brain and in the nervous system. So in the peripheral nervous system, there are Schwann cells, and in the central nervous system, oligodendrocytes that make up the myelin and, for example, speed up axonal conduction. There are microglial cells that contribute to that are like immune cells in the brain, ependymal cells that control the cerebrospinal fluid, NG2 glia that can interestingly become either oligodendrocytes or astrocytes, depending on the brain area. And then there are radial glia that support the kind of like development of neurons. And then there are astrocytes that I will more concentrate in this talk later on. They are able to sense neurons, first of all, and then also like inform back. And they also sense vascular system, as you see from this illustration, and there is very, based on the recent research, there is very intensive information exchange going on between neurons and astrocytes and vascular system. So astrocytes are involved, as I said, in many brain functions. Here is only a glimpse of them. So there are like functions that they are involved in the pre and postnatal development, like one important aspect is maturation and pruning of synapses. Then they are involved in energy metabolism and homeostasis and survival, like maintaining the extracellular ionic balance and taking up neurotransmitters. They are also involved in many neurological and neuropsychiatric diseases. But what is less known is that how they are involved in like basic sensory information processing, plasticity, learning, memory, and cognition. Basically, whether they are involved also in the computations of the brain. This will be the future topic. Astrocytes, first of all, have more complex morphologies than neurons. And this may come as a surprise to some of you, but clearly this has been a big surprise to many of us scientists as well in this field over the past years. So if you look at the cortical pyramidal neurons from layer 3, for example, from mouse, monkey, and human, you see an increase in size and complexity of these pyramidal cells from mouse to human. And if you particularly look at the the astrocytes from mouse and human, you see a large, if not to say huge, increase in size and complexity of astrocytes when you go from mouse to human brain. And this is something that has become available to our knowledge only through the recently developed imaging and image reconstruction techniques that have been developed for this field. So we didn't before understand that, for example, in vivo, the astrocytes are so ramified, so they have so many fine structures that they send their processes to reach, like the neuronal and the vascular system. So astrocytes are also morphologically very different depending on the brain area. So here are two prototypical astrocytes. The first one is the one you already saw on the previous slide, the protoplasmic astrocyte, and it's in the gray matter, and processes can extend to some 150 micrometers. Then there is other kind of large group of astrocytes named fibrous astrocytes that are more in the white matter, and they extend up to 300 micrometer. If you look at the astrocyte in a tissue, in a reconstructed tissue, you see a very close, tight interaction of the green astrocyte and its processes with the neuronal dendrite marked with gray color and with the vascular system marked with a red color in this image reconstructed by Corrado Calli in Pierre Mäki-Stretti's lab. So there is indeed a very tight relationship between these elements in the brain. And when we are trying to kind of think from the mechanistic perspective, like how do these different components interact, you see these very complex pictures that emerge. So there are really complex mechanisms and signaling pathways in astrocytes that seem to be involved in communicating both with the neuronal part and the vascular part. And I would like to point out two important homeostatic mechanisms, namely the potassium buffering and the glutamate sensing that I already mentioned. So it's very important to take the excessive potassium out of the extracellular space in order not to run the neuronal information transmission into overload, like epilepsy, basically. And it's also important to take extra glutamate from the synaptic left and transport it as a glutamine to presynaptic terminals so that you always have extra reservoir of glutamate to be released and a synapse ready for synaptic transmission, information transmission in a synapse. What is interesting with this complexity that almost all these mechanisms and signaling pathways seem to one way or another be linked with calcium signal in astrocytes, soma and processes. And this is something we look at next. So already as early as 1990, we were able to measure this kind of calcium activity in vitro cell cultures in astrocytes. And I would like to point out, as the title says, that the astrocytes don't have the sodium-driven action potentials like we knew from the early work with neurons, but instead they have calcium activity in vitro. And this spike that you see here is because of calcium, and most likely, and actually shown by our earlier modeling work 10 years ago, it's like a complex process between cell membrane calcium ion channels and then the intracellular calcium mechanisms. And these calcium signals also oscillate. And what we are doing in computational modeling of astrocytes, we try to understand, like, where does this calcium signal come from? If you look at the astrocytes with other fluorescent probe techniques like fluor3 or AM, so you see this kind of spread of calcium activity when you stimulate astrocytes. So there is like a spread from soma to processes. And this is early work. This is like 20, 30 years ago. But now what we are able to do is that we are able to also measure these things, the this calcium activity in astrocytes in vivo and in slice. And here is an example from Min and Nevian 2012 of 2-photon time-lapse imaging of calcium transients in astrocyte processes and how it develops with time when you use the type of pairing protocol for STDP that Ausra was explaining in the previous lecture. So I would like to conclude this very short introduction to glial and astrocyte biology and neuron-glia vascular interactions and point out 4 important things that I would like you to remember. So astrocyte morphology is more complex than the neuron morphology. Astrocytic calcium signals vary a lot and depend on the brain area, cell type, and developmental stage. And then thirdly, astrocyte calcium signal is involved in many brain functions. And what is of interest is whether it is also involved in brain computations. And then fourth, constant interaction between neurons and astrocytes and also with the vascular system is orchestrated via complex signaling mechanisms. And this is all work that the gliobiologists within the neuroscience have shown. But in many times this gets forgotten because it's a bit like a separate community. So those who study neurons are a separate community, and then those who study glia are another community. So next we are going to look at the modeling of astrocytes and modeling of neuron-astrocyte interactions in synaptic plasticity. And this is work together with my colleagues Tiina Manninen and Ausra Saudarkinen. When we are modeling biochemical systems, including the systems like in astrocytes and these mechanisms and signaling pathways that I was shown earlier in my talk, we are either considering a well-mixed system where we basically look at concentrations of molecules, or then a spatiotemporal system where we look at the reaction diffusion system. And in this simple case with well-mixed systems, we typically use ordinary differential equations or stochastic versions of these equations, sometimes chemical mastery equation. And then during the tutorials, you will get an exposure to a new tool in our field called STEPS that is able to do this spatiotemporal reaction diffusion modeling. I will take a short example how to model astrocyte calcium. So consider that you have this astrocyte process, you have cell membrane and then the cytosol and the endoplasmic reticulum that we are known to be like a big reservoir of calcium ions. So if we make an assumption that our calcium signal is contributed by these 3 elements on the astrocyte process, namely the leak current on the ER cell membrane, the Serca pump that is transporting calcium into the cell, and then the IP3 receptor that is quickly like releasing calcium for the astrocyte to use for information transmission. So we can use this kind of equations like shown on the right side. So we can compute the time evolution of calcium concentration inside this astrocyte process using these 3 different terms. So first the term for IP3 receptor. So just to put it in a short, this is electrophysiology recording from the IP3 channel and the kinetics is reconstructed. So this is data-based model. Then we have a term for the leak current and then another 3rd term for the Serca pump. And this is how we kind of can compute the evolution of the calcium in the cell. Of course, in reality, it's much more complex. So this is a very simple example, but it gives you an idea how this modeling of calcium in astrocytes is done currently. We have evaluated models of astrocytes also very intensively in my research group. And you have already learned that there are thousands of neuron models, but based on our survey, we found like hundreds of astrocytes models. That was more than we expected. But what actually came as a bit surprise to us is that when we were mapping the origin of these astrocyte models from the literature, we could actually see that they stem from basically 2 different types of models. So here, the same color in this figure on right means that all these publications or illustrations, they basically use exactly the same model that the earlier model used. Only the green ones are kind of their own models that build something new on top of the existing knowledge. So most of these hundreds of astrocyte models actually use exactly the same model, and they try to explain different phenomena like excitability in the brain, information transmission, plasticity, and so on. All this basically tells us that the field is very immature and we need better astrocyte models. If you are interested in looking at the state of the art, you can look at these 3 publications that we have published in the recent years. So lately, we've been also interested in understanding the role of astrocytes in synaptic plasticity in somatosensory cortex when the rodent is whisking and learning things. So in 2012, we got the first evidence of about the role of astrocytes in long-term plasticity in developing somatosensory cortex, and namely in layer 4 to layer 2-3 synapse. So Minna and Nevian did experiments, electrophysiology and STDP pairing protocol for pyramidal cell. So they were recording from a pyramidal cell and assessed the synapse functions. And they also were recording from cortical layer 2-3 astrocytes, so the nearby astrocyte, and also used calcium imaging, like in this lower illustration. What goes on when you induce plasticity protocol, and then how the astrocytes behave. And they did a set of complex experiments with pharmacology, and basically they were able to show that this phenomena without astrocytes, it doesn't happen. So this was one of the first evidence that astrocytes are at least during development and heavily involved in computations in the brain. And we set up a goal. We wanted to understand how astrocytes contribute to this. So first we made like a very phenomenological model of this system, but with Ausa-Saudarkiene, but then quickly realized that we really have to make this kind of mechanistic model in order to understand what is going on in there. And we were joined by Tiina Manninen, and we developed this model where we have one compartmental presynaptic layer 4 spiny tellate cell. Then we have 2 compartmental, basically soma and dendrite, postsynaptic layer 2-3 pyramidal cell, and then 1 compartmental astrocyte fine process. And we incorporated basically all the evidence that has been shown, like all the key elements, like you see in this figure, you see these different molecular players, biophysical properties, first of all, molecular players that has been implicated in the literature to be contributing to long-term plasticity in this synapse. And to make the story very short, we had to go through 70 references in order to find the molecular players of this synapse. And we ended up with a model that has 80 differential equations and 170 parameters. And on this slide, you see a glimpse of the equations, like on the right side, the equations for the presynaptic terminal, and then the parameters. And as you see here, we indicate the references where we have taken the parameter values of this model. So this is very biology-based model. And we didn't guess many of these parameters, maybe a few, 1 or 2. To kind of narrow down, so basically we use these 70 references to build up our assumptions, like what contributes to this phenomena that Minna and Nevian were seeing. And we could point out 3 key assumptions behind the model. So the first key assumption was that in this developmental phase, in this specific synapse, a postsynaptically released endocannabinoid 2-HC is important, and it's released from the presynaptic terminal. It activates the calcium in astrocyte. And then a second key issue for this phenomena to happen is the astrocytic calcium-dependent glutamate exocytosis. And glutamate activates NMDA receptors on the presynaptic neuron. And then the third key element is the calcineurin, which we also model, which links the astrocyte to the presynaptic terminal release of glutamate. So we sort of describe with this model this kind of loop between these three components. What we were able to predict with this model is that you all learned from Ausa Saudarkene this STDP induction protocol. So when we use like a very short temporal difference between the pre and postsynaptic and presynaptic activity, so this led to stronger TLTD in our model. And this very same, so our simulation with the black is very close to this green experimental data that is actually obtained from another validation data set, Banerjee et al. From 2014. So we use these two data sets to validate our model that this TLTD is exactly the same as in the experiments. And then I would like to point out that the model, when we built it, we used additional 70 other references to build the model up. So basically in modeling, it's important that when you build a model, it's different data that you use to validate the model. And what we could show with this model and predict that it's really the astrocyte-mediated cellular and subcellular mechanisms that seems to contribute to this phenomena, like was speculated by Banerjee and also indirectly shown by Min and Nevian. What we can do with this kind of model, we can perform lots of simulations. We can try to understand like the system. We can have a similar protocol like in experiments, like before this TLTD induction protocol, then we can induce long-term plasticity and then look at how the system behaves after this plasticity is induced. And for example, with our model, I point only one simulation here. We can see that there is reduced synaptic glutamate release when this protocol has been applied to our model. So what this model allows us is to kind of study the time-wise behavior of different state variables that we have in this model and ask questions. What happens if you have like if you, for example, don't have any more the presynaptic NMDA receptor, if it's somehow not operational in this model. So what might happen in the living in vivo system? To put the story very short, I would like to point out that this model links complex biochemical networks at the pre and postsynaptic sites and astrocytic sites to the electrophysiology and time window of TLTD induction at the vertical somatosensory layer 4, layer 2, 3 synapse during development. And the model predicts for the first time the dynamics of these molecular mechanisms underlying TLTD. And this is not possible to do in experiments, all of them at the same time, at least not. And I wanted to make a small comment at the end here and conclude like what I think this means in terms of brain computation. So I think that during TLTD, this single astrocyte acts as a delay factor for the fast neuronal activity. And basically what astrocytes do is that they mediate integration of fast sensory processing because they are all the time reading the neuronal activity with a slow non-neuronal processing. And this is the way how they modulate synaptic properties in the brain during development. And once I saw this model functional, once I understood what it's doing, I made up finally my mind that I started believing that yes, they must be involved in the brain computations. But so far, I have studied it myself only during development. So I don't say anything about adulthood. That has to be addressed separately. We provide all the model and also the appendix to explain all the equations and parameters and state variables. So you can look at it in GitHub and Model DP. Towards the end of this talk, I also wanted to mention two other projects that we are doing in addition to this synapse and synaptic plasticity modeling and astrocytes. So in the HPP EBRAINS consortium, we are developing also astrocyte neuron interaction modules for spiking networks that would allow like simulating astrocytes also with nests. And then this is work with Markus Diesmann and Sassa van Alpada and my group. And then there is another study in the HPP EBRAINS context where we model neuron-glia vascular interactions in brain signals. This is work with together with Federico Teslen and Alain Destex. And you can look at the first paper that is in bioRxiv right now from this study. And with this study, we kind of try to reach in humans from the molecular level up to the system level in the brain and explain the mechanisms behind bold fMRI signal. I point out that this field of modeling astrocytes and astrocyte neuron interactions is very much in the beginning. There are some tools that emerge right now, like the Astro tool to import the neuron astrocyte morphologies to neuron, but it's in the very beginning phase. And I think this is my final slide. So I would like to point out a few literature sources. The book, The Bible of the Neuro-glia by Helmut Kettermann and Bruce Ransom, and then the recently published first book on computational glia science, where we also contributed a chapter. And then a couple of references like review articles where you can start studying this field. But I think you will get all these slides, so you can then look at these references.